All right, I think we are ready to roll. Good afternoon, everyone. For those of you that don't know me, I'm Mindy Dwinell. I'm at the Medical College of Wisconsin. And on behalf of the TCDC, I'd like to welcome you all to the first Meet the Experts series event. This series is designed to cover topics of interest and importance to the SCGE consortium. And today marks the launch of the series, which will take place several times a year. We do welcome suggestions for future events, since our goal is really to make this series as valuable as possible to members of the consortium and then eventually to the broader scientific community. So now I'd like to turn over the virtual microphone to introduce Jean Dedashti, who has generously agreed to moderate today's session and will introduce our panelists.
Thanks, Mindy. I'm Jean Dedashti. I'm a program officer within NIH, a National Center for Advancing Translational Sciences. I'm a program officer and I work very closely with PJ Brooks. Today, I'd like to welcome Dr. Peter Marks from the US FDA. He's the director of the Center for Biologics Evaluation and Research. Thanks, Peter, for taking the time to join us today. And also Dr. PJ Brooks. He's a deputy director of the Division of Rare Diseases Research Innovation, or DRDRI, within NIH NCATS, and he's also the co-lead on the SCGE program. So thank you both for joining us. And I'll get the questions started with a question that came through last week. Is the FDA moving forward towards official recommendations on platform technologies and therapeutics? So Peter, maybe I'll start with you. And as a starting point, it might be helpful for the audience to understand how FDA, particularly CBER, defines platform technologies. And then maybe you can touch on the progress that the agency has made towards that.
Yeah, no, thanks very much. I think let me just back it up to the point of saying that, you know, platforms can mean a lot of different things to a lot of different people. I think for us, I think we are thinking of platforms, a gene therapy platform in particular, or perhaps certain vaccine platforms in particular, as being essentially not just one component of some. A platform isn't just one component, but is multiple components that go together, such that when you swap out one of those components, you can essentially use the information for all of the rest of them moving forward. Let me just say how that applies, for instance, to AAV gene therapy. The idea here is that for an AAV gene therapy, one would potentially have a given serotype of AAV, say an AAV8, have a certain size insert that you can put into it for purposes of a given platform. And then the idea would be that you would use the same manufacturing process for that vector. And that would allow you potentially to also use all of the information for manufacturing, for toxicology, for quality control for that vector, even though you would swap out into different inserts as you move along between different gene therapies. Now, we actually have this now in law as part of the Omnibus Appropriations Act of 2023. There was a platform technologies provision which allows us at FDA to have take an approved product, and then a manufacturer can use reference to that approved product as they move through kind of a platform approach. So we have a variety of ways to apply that now. And as we're thinking about it, I think it will apply pretty neatly to AAV vector gene therapies, potentially to other vector gene therapies, potentially even to in the vaccine world, for instance, in the mRNA vaccine world. It's possible that there could be some applications there, but for today, we might as well focus on the gene therapy world of AAV, where I think, and on the other place where I think we very much see this as an approach which would be in CRISPR genome editing, where there you possibly have what I would call the poster child of a platform, because you have a molecule that's largely going to be the same, no matter whether you're from disease to disease, provided you're targeting it to the same tissue with the same delivery mechanism.
Yeah, Gene, maybe I'll just follow up with that. I mean, I think that in 2019 or so, we developed the PAVE-GT program, which actually stood for Platform Vector Gene Therapies, to try to do what Peter just described there. And that's ongoing along with the BGTC program that are focused on AAV. I think one thing I've learned from doing this, and maybe Peter learned or already realized this, is that it's not quite as simple as it seems, popping a different gene into the same AAV vector backbone. I even, had a diagram where there's like a shipping box, and there's a few problems with that, which is that the stuff that goes into the shipping box isn't always exactly the same or what you think it is. And it does change, in a sense, the shipping box when you put different AAV, different genes in there. Whereas I think the potential benefit for gene editing is you have really three components: an editor, a delivery system, and if it's CRISPR based, a guide RNA. And you can use, some of those would be exactly the same, and that can be, I think, a more rational basis of a platform. And I think, Peter's idea of the poster child, I think that's why that is. So in some ways, it's almost a better example of a platform than AAV.
Thank you both. I can't see any questions, but I guess if questions come through, Mindy or Angela, you can let us know, or we can circle back on this. I'd like to move on to Dr. Fyodor Ornov has requested that he ask a question. He's going to propose a scenario, and this actually is something that came up in a recent SCGE Interact meeting. So I don't know, Fyodor, if we can include him on the panel, that he can actually ask this question, present the scenario, and ask his question directly to the panelists.
Yep, he has been elevated. Should work.
Thanks, Min.
I have been elevated. Can you folks see me and hear me?
Yep.
Peter, thank you very much for making the time. Really appreciate it. Good to see you. I really want to honor my SCG colleagues. First of all, thanks PJ for gathering us here, and SCG leadership. And my colleagues in this particular instantiation are Kiran Musunuru, who's a physician scientist at Penn Medicine, and Rebecca Ahrens-Nicholas at CHOP, and also Chris Saha at University of Wisconsin, who, among others on the SCG program, are facing the same minimum viable product of CRISPR as a platform scenario. You have a newborn child with an aggressive, debilitating, fatal genetic disease for which the hospital where the child is diagnosed, it could be Penn Medicine, CHOP, it could be UCSF, it could be an inborn error of the urea cycle, or in our case with Michelle Hermiston, Peter, I know you know her well, this could be a severe case of familial hemophagocytic lymphohistiocytosis. The children have to go to a transplant in the first 2-3 months, or else it's just terrible. And the transplants are typically bad and lifetime GVHD. We have an open IND for using one gene editor and one guide RNA for one specific mutation in a cell type. The manufacturing process is locked and loaded. The patient coming in has a new mutation. We have the ability technically to manufacture a guide RNA specific to that new mutation while anchoring everything else the same, whether ex vivo manufacture of gene-edited HSPCs, or in the case of Kieran and Rebecca, LNP to edit the liver. We would love to get a sense, Peter, of your thinking of what framing could exist in terms of, having the regulatory path of with that brand new guide RNA where everything else is the same. what regulatory process could we start thinking about where the length of that process is commensurate with the lifespan of the patient? I know this is a difficult question, so I'm sorry to like throw you such a fastball with a curve on it, but like here we are facing the same pitch.
Right. So I guess what we're trying to, I guess to summarize, the concept is, how can we kind of bootstrap more rapidly without having to go through essentially the pre-work over again. I think that's right, Fyodor. That's pretty much what I'm hearing here, and I think for the reason why, and I think actually PJ set this up pretty well. I was a little, I don't like to put down AAV unnecessarily. It's an important vector system, but I think that, with genome editing, our thought is that this will be a place where once you show, I think the idea here is for a genome editor, that once you show that you're correcting something with one construct, I think we're not going to ask people to go and show that that we're going to need data for every possible nucleotide or set of base pairs that are corrected. It's going to be, the principle is that you can correct this gene using the construct, and provided we have data on the off-target analysis, I think we're going to try to figure out a way to expedite going from for the treatment of an entire disease entity, so that we're not developing 18 products for one rare disease. You're developing one product, and my guess is that we'd require the description around one of the mutations, right, and the rest would essentially be included by the principle that you're doing genome correction, because we can't it'll slow things down to a crawl, right? I mean, and I think the idea is that a corrected gene will work, right? And so as long as we're confident that the construct that the editor is correcting that, I think that's where we're going to go. Is that answering your question, Fyodor?
It does. So I want to read back what you said, and then PJ, if you agree, and Jean, I'd love to, if Kieran Musunura or Rebecca Arns-Nicholas or Chris Saha are on the line, I'd love to have them join this, because again, I'm grateful to them for their partnership on this. Peter, reading back to what you're saying, the analysis of on-target potency that the new editor we're bringing in is comparably effective at repairing that mutation in that same gene as the one we have the IND for, and providing a robust off-target package for the new guide, would be sort of the minimum viable product for us to think about as we're, again, in a scenario where, we have a child with, really severe disability unless the treatment. Thank you, I appreciate that. Chris, it looks like you are here, so, if there's some way to bring you online, if PG, and thank you, Peter. This is terrific news. Thank you.
Yeah, no, I think we're on board with trying to solve this problem, right? Because I think your concern, I think, is the one that I think we're having, right? Which is that you're going to have, otherwise you're going to have each new child will be a new discovery, right? And I think what we need to do is find a way to leverage the information here, so that what you're doing is essentially showing that in principle you can correct a given feature. Now, I think it may be that there may be stretches. I don't want to say that it's going to be okay to do this in every case, because it may be that as we get more audacious with what we're doing with genome editing, we may need more data. But for the simple kinds of genome edits that I think you're talking about for in these situations, I think we should be able to bootstrap our way through here. PJ, do you want to comment at all?
No, I think this is really a regulatory question, Peter, and I think we're all very interested and happy to hear your perspective.
Yeah, no,
I don't want
I
to.
think
I don't
so.
want to
So
beat up.
our
Yeah.
our goal here, Fyodor, is to try to simplify this. And I think we'd rather spend the time thinking about understanding the off-target effects, right? Potentially, than spending a lot of time of redoing all of the toxicology and manufacturing information that's just going to be redundant.
Thank you. I really appreciate this. Chris?
Great. Want to share the enthusiasm from Fyodor and PJ for sharing your time with us here, Peter. I was following up on this bootstrapping effort and to see whether you think there's a category of diseases where the severity matters. For us, we're targeting the eye, and some may consider that not to be life-threatening per se in the same way that Fyodor mentioned for the liver and blood programs. Do you think this type of simplification and bootstrapping needs to be only applied to scenarios that are, imminently morbid, like have very bad outcomes? Or do you think that this should be, for some variants that are maybe a bit more mild, still a valid way to go?
Yeah, so I think probably like many things, we may want to get our feet wet in the worst case scenario first. And once we see that that's working well, I think it would be pretty simple then to move into the less serious cases. It also may be that it may be simpler with certain things where we're doing where mainly what you're doing is fixing single nucleotides or different single nucleotides. Maybe easier to bootstrap to than when you're needing to stretch different stretches of DNA in different in different areas of the genome for a given genetic disease. So I think we may start. It may be easiest to start in the most serious cases because there's just we have more tolerance for risk. I'm not sure how long we're gonna. It's not like I don't think that's gonna last for years while we're doing that. I think what we need to do is because this is all somewhat new. I think we'd want to feel like in a serious disease we could get some, a serious disease or two, we could get some good experience seeing how this type of approach allowed us to cover. What you're really talking about is more of it's a process is the product, right? It's a process of what you're going to do to show that you're not going to have off-target effects and to produce the product and have correction rather than just what the end product is, right? In some ways, what we would be doing is somewhat akin to what we do with some of our personalized vaccines, right? It's something where you the process here of how you get there in terms of showing that you don't have off-target effects and that you're likely to correct is where the money's going to be.
Peter,
Does that make sense?
Yeah, could you maybe expand a little bit on the personalized? I think there's a personalized cancer vaccines, in case everybody's not aware of that, because I think that's a really interesting analogy, right? That every different patients have different tumors, so you need a different target to generate the vaccine towards, but it's basically making messenger RNA. So maybe you could talk a little bit more about that so people understand the analogy.
Yeah, no, thanks. So the idea here is that we have some products where the by and large the product, there's a process that's the same, right? It's the same process that's used to make the product, but at the end of the day, what comes out, the expression of the cells, the essentially the antigens or a piece of the product is different from depending on an individual patient's input. And so I think the idea here is that this paradigm for having a process that leads to products that could be different yet do the same thing ultimately it's not a totally new one for us at all.
Are each one of those new INDs, or how do you handle that? I'm just thinking about the analogy that we can gain
No,
here.
it's the IND here. These would be I think the idea here, and again, I some of this is all new territory, but I think the idea here would be you would think about this as the IND would be, I think, for a CRISPR construct that corrects the gene in disease X, and encompassed in that IND would be all of the different, so like, I think that's what you're looking for, right? Because that's what we'd be looking for. So we don't have, otherwise you would needlessly have 94 INDs sometimes where they're all going to be doing the same thing ultimately. And so I think instead you just have it, this is an IND for a CRISPR construct that corrects the abnormalities in, MPS 2 or in GM1, depend, whatever the disease entity is, and that you would encompass in that all of the constructs. Now, I guess, could it get complicated? For instance, in certain genetic diseases, you have a combination of point mutations, large deletions, inversions, insertions. I think that's some of the nuance we'd have to sort out. But the ultimate goal would be to keep it as simple as possible in terms of the regulatory paperwork.
I'm a Beatles fan, and this is better than Sgt. Pepper to my ears.
No, but I think even there, I think that the goal is to have a corrected gene at the end of the day. I think that's the IND is to get a corrected gene, right? I think that's the way we would want to think of it.
Rebecca?
I had a quick follow-up question actually to that point, and I want to echo everyone's gratitude for this discussion. I think it's really important. I guess my first follow-up question real quick is that to add other variants then, once that IND were open, would you envision that sort of as an amendment with the off-target data for that particular variant of interest for that gene, for whatever IND is already open, or what would that look like sort of practically?
Well, I think what you would do is, as part of the process, you would be doing for each one of these, you would add into the IND, you just submit to the IND the off-target analysis that was done for that particular construct, right? I mean, it would become how we could figure out how you would do it on an ongoing basis, but you'd basically be doing what would probably be a combination of an in silico and perhaps some in vitro in living cell analysis to that complementary analysis to see what your off-target effect is, file that and go. Do you know what I mean? Something like this.
That sounds amazing, and I think that is an agreement with a lot of what we have been speaking about and hoping for. So thank you for that openness. I think my more direct question was, in our case, the most serious cases that we were referring to are essentially all infants. And I know oftentimes the FDA would like us to start in our older patient populations. I guess, how do you feel about starting with this type of work in an infantile population?
Yeah, well, I think there has to be, we have to have this concept of prospect of direct benefit. But and that's why I'm saying you start in if you're talking about a really bad disease, I think there's a fair amount of openness towards what would qualify there. And I think, again, we can, that's part of the power of CRISPR, right? This concept is that I think is if we have, a good handle on the data supporting that this approach could work from some model system or from some other situation, I think it's probably a way we can get there. And I'm increasingly feeling like we have to be a little bit more bold. Again, obviously with appropriate, appropriate input from IRBs, et cetera. But appropriately bold in where we're trying to use gene therapy here, so that it actually brings benefit. Because in some of these diseases, once you lose function, that's it. You're not getting that function back. You lose the neurons are not coming back.
Yeah, thank you.
So I think just to finish up on that, I think that's where a discussion, coming in and having a discussion with the agency so that our reviewers understand the urgent nature of the situation, and the potential for the intervention at an early time to prevent irreversible harm that will occur otherwise. I think that can help us here.
That sounds very reasonable. Thanks.
Thanks, everyone. Any other questions for Peter or any discussion points here before we move on to the next question?
I think Kieran might have had one.
Kieran, oh, he's not on. Mindy, could you please make Kieran a oh, there we go.
On his way.
Thank you.
Yeah, I actually did have two sort of related questions. Thank you so much, Peter, for giving us this opportunity. Except my questions disappeared. So I had them all written out, but okay. So first question, based on what Fyodor just said, the scenarios that he laid out, as well as feedback we've received recently in an Interact meeting, as well as some discussion on a public webinar hosted by the FDA just a few weeks ago. The scenario that was laid out for us is that if we had two very similar gene editing drug products, oh, and here it is, okay. Good, two gene editing drug products differ only by a guide RNA. They're both for the same disease, treating two different pathogenic variants. The guidance we received was it should proceed as follows: file an IND for the first drug product, obtain clinical data on that drug product, and then file an amendment to the IND covering the second drug product. And so the first clarification that would be good to have is how important and how much clinical data would we need to get before there would be a comfort level to allow us to go ahead and file an amendment for the second drug product, and maybe a third, a fourth, and fifth, and so forth. And then to extend upon that, here we're all just talking about one disease, one gene, but if you're talking about drug products intended for similar but not identical diseases, for example, two different inborn errors of metabolism that are both centered in the liver, like say two urea cycle disorders, they affect the same pathway, the clinical endpoint's the same, but they are technically two different genes, two different enzymes, two different diseases, formally speaking. Would the same paradigm hold, or do you envision that really applying to one disease, and then once you jump to another disease, that becomes a whole new ball of wax, and the expectation would be to file a fresh IND covering that disease? Sorry if that was a little discursive.
I think we lost Peter.
Uh-oh.
Uh-oh.
Oh, here he is. Okay, great.
I don't know if I have to go through that whole line of questioning again.
No, I heard most of it, and unfortunately then our internet just died and came back on, so sorry about that. So I think, so the question was how much clinical data you need, and I think the question is, first of all, filing an amendment to an IND should not be a big deal. That's just to get the guide information for the second piece. How much clinical data do you need before you can move on? I think is something that I think is something you'll need to, it will depend on what we're looking at as the outcome measure to see whether we think you've gotten appropriate correction and what you can show us. It may be very early information that if you have an absent protein, could it be something as simple as expression data from essentially a northern blot equivalent maybe it could be. But obviously we'd love to see, I think it might be the ability to see protein expression, or I think it does not necessarily, though, have to be clinical data per se before you move along. And I think you'd want to have that conversation with us, because again, certain diseases it's easier in than other diseases.
Right.
I think what I hear what you're getting at with this is that, are you going to have to wait a year and a half before
Yeah,
you come back in to move to your next? And I think that's not what we're looking for. I think what we're looking for here is reasonable assurance that we can see some hint that things have worked, and I think that's the one of the things that I just should say it here. Some people are wondering why it's easier in this world of genetic medicine than it is in traditional small molecules. Well, it's easier because we can use, technologies, be they blotting, Western blotting, Northern blotting, or similar, to actually show that we have expression of something that you didn't have expression of before. It's so, and so you have mechanism built into the approach, which you just often don't have when you're doing small molecules. So I think I would lean heavily on this, and I think our folks are willing to lean on this some. And again, I think you may see us. We will be putting out some guidance on platform therapies. We're mandated to do so by the law, and in the not too distant future. But probably in the future, we'll have to put out additional guidance as it applies to CRISPR, because otherwise it's going to be all of your questions basically need to be collected in a document so that everyone has the benefit of working from the same playbook.
Oh, that's great. That's really helpful. And the second part of my question, which you might have missed because of the Wi-Fi dropout. And so everything we've been talking about right now are different drug products for variants in the same gene. But let's say you have two different genes, similar but not identical diseases, right? So let's say like two different enzymes in the same pathway, two urea cycle disorders, two different inborn errors of metabolism, both centered in the liver. So they're related diseases, but they're not exactly the same diseases. Would the same sort of thinking apply, that as long as the clinical endpoint is largely the same, we could go through the same amendment process? Or are we really talking
So
on a disease by disease basis?
So Karen, I think we're thinking through this. I think the idea of kind of a pathway approach, which is that if you could measure the same outcome or measure the same metabolite, because in some cases, right, you have, if you have the same metabolite that you're watching, for instance, heparin sulfate, you can imagine that you could have a variety of different, you could be correcting different genetic defects in what are technically now different disease entities, and still try to be looking at one endpoint. This is something we're discussing more about how best to, and so I don't want to give you something that's fully cooked,
Sure.
and that's when it's not. But it is something we're thinking about how we can again help move this forward without having to go back and reinvent the wheel. It may be that we'll have ways of thinking about how we can group endpoints for diseases that are related. But it's actually new territory. It's cool to be thinking about. It's new territory. But I think we do have to figure out ways to lean in to be able to do this again, because we want to be able to apply this technology as broadly as we can, and take calculated risk, not that will lead to hopefully reproducibly good safety and efficacy in the application of the technology.
No, thanks. And it's really great to hear that you're thinking about and actively engaged in that.
I'm sorry that we don't have some of this. I wish some of this is kind of it's baking the cake. We're still mixing the batter. But we realize what the ingredients I think need to be.
Right. So I do have one more question. I don't know if like I should let others take turns and just be quiet now, or should I just go with it?
I think, Karen, if it's related, go for it.
Yeah, okay. So it is sort of related. So, we're mindful of what happened with Melicin, the first N of 1 antisense oligonucleotide treatment, several years ago. And our understanding from what has been published is that that proceeded to investigational treatment under an expanded access IND application. And so naturally, we're curious, would the agency be open to an expanded access IND application for an N of 1 gene editing therapy in a scenario, of course, where, the patient has a limited lifespan, terrible quality of life if they don't receive a treatment? Is that something we could be thinking about, or is the preference really to go more the platform approach?
Yeah, I think in a serious situation, this is a single patient. It would be a single patient IND, emergency IND, or single patient IND. I think we'd be open to consideration, especially as we're getting this stood up. So I think, again, I think if you come up with a situation like this, I would contact us sooner rather than later to have the discussion, because my guess is once we get some of these INDs stood up, it'll be easier to put them in there. But we never want to, we always want to try to do what's right for the patient now. If there are instances that come up now, I think, again, provided we understand the urgency, I think I think it's reasonable to do a single patient IND.
Great. Thank you so much for your indulgence.
You're welcome.
Appreciate it.
So we do have questions in the chat, and Sonia also has her hand up. Should we have Sonia? Okay, I'll unmute Sonia. Oh, you're already. Sonia,
Am I unmuted? Okay.
Yep.
Hi. Hi, Dr. Marks. It's terrific to be able to speak with you today. I wanted to take us back to the theme that you can't get those neurons back. And to give a little background on my team, we're working on genetic prion disease, rapidly fatal neurodegenerative disease, always fatal, very, very rapid. And, as we think about bringing forward a gene therapy for this disease, I think we're walking a delicate line, because with an irreversible therapy, and our project will use a novel AAV vector, I think it will make sense to go first into a small number of symptomatic patients. And as we think about what that's going to look like, we're also, and hopefully, we'll be in to speak with the agency about this soon, because I think it's a detailed discussion, but we're also imagining, how do we design those studies from first principles so that we can learn from those first severely impaired symptomatic patients as much as possible about whether this therapy can be taken into people whose genetic risk puts them at close to 100%, certainty of the same fate. But if we wait for them to manifest symptoms, it'll be too late. So this is sort of your paradigmatic lightning strike. And, my husband and I have been leading a natural history study looking for prodromal changes in the at-risk individuals for many years. And so far, nothing has undone my assumption that this is basically a lightning strike. I think as we go into these symptomatic patients, we will want to learn everything we can about target engagement and safety, understanding that neither will be unconfounded. But how to sort of have the right sort of preemptive discussion so that we can learn as much as possible about the potential for swift advancement into pre-symptomatic people where we know this is coming for them. And, this is tedious for those who know me, but I'll just, say to those who I don't know, I'm one of those people. Today is my 40th birthday. This is coming for me. So we ask from that place.
Yeah, so point well understood. I think this is where this is a particular one where having the conversation with the agencies are a really good one, because I think what we would probably be wanting to look for is I think you already said the key word, some idea that we are engaging the target appropriately. Do you see what I'm saying? That we're
Yeah.
actually having some effect. I think I get it, right? You're trying to see a clinical efficacy here, maybe is probably.
Oh, I think you
Other
froze,
people still
Peter.
hear Peter?
I think he froze.
Okay.
So hopefully he'll unfreeze. Happy birthday, Sonia.
Thank you. I'm leaning into celebrating it. It comes with mixed emotions.
Oh,
I would think so.
All right, we've lost him. Hopefully he can rejoin quickly.
I mean, so far, so very, very good, if I may say
Yeah.
so. We just heard,
Theodore, Becca, Kieran, are you guys okay to go back to attendee mode, or would you like to hang out here? Okay. I didn't want to insult anyone, sorry.
Oh, here he comes. Nope. Yeah.
I'm sorry. It seems like unfortunately I've had stable internet all day until they decided to go. Apparently they're working on the internet in the area. So I apologize. I'm so sorry. So I really appreciate that last question. And I think it's a matter of coming in. Did that make sense? The answer
The
there? I
target
think
engagement, yes.
yeah, I think we really would want to discuss. I think our folks, I think Nicole Verdun, who's the head of the Office of Therapeutic Products, I think she's talked about this, that I think in this case, this is where this concept of using some marker, right? Some biomarker, something that we can say that we can make the connection, even if it's not a perfect connection, but we can make the connection that we see this change here, we see it somewhere else, and ergo, it's likely if we take this into the earlier setting and we can make that same change, we will prevent the outcome that is an adverse one.
That makes sense and is encouraging. Thank you.
You're welcome.
Great, thanks, Peter. Thanks, Sonia, for your question. I think we'll move on then to some of the questions that we received last week. And these questions are more focused on IND enabling studies. So a question that we receive is, what are the expectations for non-clinical pharmacology and toxicology studies to support an IND for gene editor comprising a single protein nuclease scaffold and different guide RNAs to target different mutations within a single gene? And PJ, maybe we could start with you on this question. And if you could touch on your learnings and what you've been hearing across the various NIH gene therapy programs that you've been leading, what you envision as minimum requirements. There's a lot of discussion around what's the minimum requirement for IND enabling pharmacology, toxicology studies to support an IND for perhaps a gene editor comprising a single protein nuclease scaffold with different algorithms.
I can only say that I think that that's something we're actively trying to determine as part of the BGTC, is whether we can come up with that and ultimately make that available to the public. Obviously, the way you find out the real answer is through our interactions with the FDA in these formal meetings, and so that's how we're proposing to go about it. So I don't think I would like to opine any more about that, except to say that I think it's obviously a very important question given the costs of some of these approaches, and particularly the non-human primates. But beyond that, I don't think I want to step too much. I'd like to let Peter answer that one.
Yeah, I feel like we've we've done a fair amount of answer. I mean, I think I'm trying to figure out what's uniquely different about this. Can you just repeat? I'm trying to figure out what's uniquely different from some of these previous.
I think you did touch on it, where for each of these different guide RNAs, as long as it's the same single protein nucleus scaffold, targeting a different mutation within a single gene, that you wouldn't necessarily have an independent tox package for each of these.
Right,
So
right.
you did sort of touch
Right.
on
No, I think the idea here would be to have a tox package that supported what we'd almost it's like the exemplar, the initial product. One of them you'd have to have a tox package on, but then as you move through your different guides, you wouldn't repeat that tox package.
Yeah, and I might just say, I feel bad. I sort of beat up on AAV at the beginning that, I talked about for gene editing, you need an editor, a delivery system, and a guide RNA, but the editor and delivery system could be part of AAV. And then if those things are exactly the same, then the new component is a guide RNA, and that could be part of this amended IND application that Peter said. So I apologize to all my friends out there if it seemed like I was beating up too much on AAV.
Yeah, I mean, I think just so you know, I'm trying not to beat up too much on AAV. I have to be honest that when you look at the potential for CRISPR plus lipid nanoparticles plus some of the targeting that people are starting to be able to do, it's very tempting to beat up on AAV, but AAV is what we've got right now, and it's what's powering some of the work. So I think we have to be kind to AAV, even though it may be retired at some point, or move into retirement planning.
Thanks, Peter. Thanks, PJ. The next question is related to IND enabling studies and the requirements around non-human primates. So the question is, and we received this last week as well, can you comment on what the approval IND safe to proceed process would look like for a gene therapy that uses a delivery vector that is human specific and does not engage the target in non-human primates or any other non-human species? That's i.e. preclinical tests for efficacy can only be done in humanized mice, but not non-human primates.
Right. So I think we would be thinking about, I think this is where you come in and have a conversation, because we're not going to ask you, non-human primates right now are scarce enough, as people may be aware, that we're not going to ask people to do studies that don't actually bring informative results in non-human primates. I think, though, we do have human organoids and we do have humanized mice. And so it's possible that we could ask someone to do studies in those areas. And that would depend on a discussion with the review team. It would depend on what we know about the constructs that's being used, and what we know about what an analogous construct that may have been targeted for, because you can obviously use if you have a non-human primate targeting construct, it can be used in non-human primates to show proof of concept. So it would really depend on the totality of the evidence that we had here. But I think we agree that just doing toxicology studies with something that really can't show you the primary toxicology, right? It could show you that some impurity in your preparation, harms the non-human primate, but it's not going to show the primary toxicity. We're not going to ask people to do that.
Thanks, Peter. PJ, do you have anything to add to the discussion?
No,
All
I think I'm just glad to
Yeah.
hear Peter's answer. I think it's an interesting question about what some of these toxicity studies can tell us. And if it's a matter of concern about something acutely toxic in the preparation, that might well be detected in mice and rats. And I think that's kind of the idea of using them where possible, right?
Great, thanks, PJ.
Exactly.
Maybe, Mindy, are there any questions from the audience that we could touch on now?
There are, and Kat Lutz had her hand up and would like to ask a follow-up question on de novo mutations. So Kat, are you
Sure.
available to unmute? Let's see. See. Oh, I might have muted you.
Yep, I think I have it. Can you
Okay,
hear me?
good, perfect.
Thanks so much. I do appreciate the opportunity to ask these questions. So I actually have two questions. One is to just summarize a little. You know, I think we're all dealing, or some of us are dealing with pediatric populations that are ends of fuse and their de novo mutations. And it's sometimes hard to predict how those patients will manifest because these are very heterogeneous populations. So one of the hopes, and I think I heard you say this, and I don't want to put words in your mouth, but I just want to say it back to you so I can make sure that I'm, you know, also advising our foundations and patients and parents appropriately. That if we were to do a proof of concept with a particular de novo mutation, with the editing strategy, and provided that the manufacturing and other components were the same, and the only things that changed were the guide, that we wouldn't necessarily have to have, you know, separate humanized mouse models, separate human studies, and that could serve as the proof of concept, if you will, so that we can then use that editing strategy to work on different patient mutations within that gene. Is that a fair?
Yeah, I think that's what you heard, because the idea would be you'd show proof of concept. And the idea is that that you would assume that that you essentially have a host of similar similar constructs. They're going to differ in the guides, but the large bulk of the CRISPR is going to be the same, right? And the guide's going to be different. You're going to characterize the off-target effects of the guide. And so I think we're not going to need to go and do all the preclinical studies over and over again for each one. It's a waste of resources, right? It's better off focus on trying to ensure off-target effects and ensure that you're actually getting a readout that you can understand from this. So I think that's where we'd rather focus the effort. Have I answered? Maybe I misunderstood something.
No, it does. And I would be happy for you to put me out of business from the preclinical standpoint, right? That would be great if we didn't have to continue to work with all of these different, by engineering all these different mutations in the mice. It's hard to know what's going to be applicable. My other follow-up question was just around the cost and the toxicology. So we certainly hear, and I did hear your question on the non-human primates, but I also want just a little bit of clarity around whether mice and rats would be considered sufficient from the FDA in terms of toxicity and tolerability.
Yeah, so there, I just want to, I'd say, come in and have a conversation, because it would depend on the novelty of the application, what's being done. I think we try to be species appropriate, using the least precious species. I don't know, I hate to be judgmental, but the least precious species possible. So I would have a conversation.
Thank you.
Again, I think we're not wedded to non-human primates when we can avoid them.
And would you consider, though, mice as a species in lieu of rats for these types of toxicology components?
Yeah. Yes.
Thank you.
Smaller rodents rather than larger rodents. Yes.
Thanks, Peter. Thanks, Kat. PJ, do you want to add anything to?
No, I think those are some of the questions that we're trying to sort of get at with the gene therapy field is when, under what circumstances can we do the toxicology studies in only rodents? And I think a lot of that will apply to the genome editing studies as well.
Thanks. I think we're at 4:59. Do we have time for another question? a thought-provoking one. PJ, this is your question. So I don't know if you want to go ahead and ask it and start the
Yeah,
discussion.
it's kind of like a forward-thinking question, and if you know, what can we imagine looking into the future would be the optimal state for genome editing to make it available to as many people as possible, looking a ways down the line. And one thing I've often thought about is, could we ever imagine where genome editing would sort of become the practice of medicine, somewhat like liver transplants or stem cell transplants, realizing that, you know, there is this drug-like component, but could we ever get to that point where it becomes like the practice of medicine? You go into the hospital, there would be the Department of Interventional Genomics, and Petros is on here, I think, coined that term. Could we ever imagine getting to that point, Peter? And if so, how? How's that for a question?
Yeah, no, thanks. I appreciate that you gave me at least a half an hour heads up on starting to think about this one. This is I don't think it's as crazy a question as it sounds, because if you have, I mean, I think the issue, I guess, would be you'd have to have a combination of how you're going to make your CRISPR and how you're going to deliver it to the right location, right? But I guess given that you could pick one from column A and one from column B to get it to the right place, right? Could you imagine that for things that need to be targeted to the liver, that you could imagine that we got there to this being, you just type in the sequence or it gets downloaded into the machine that makes the CRISPR in your hospital's laboratory, and then you go and administer it. Although it sounds like science fiction, I think probably what you'd want to see is that repeatedly in different diseases with different constructs, we continually get the same result without novel toxicities, which is that you would go through an algorithm of the computer would design a construct. That construct would be checked for off-target effects, and it would be it would be formulated into a delivery system. I guess if that's the idea, to have that done locally, you'd probably just want to know that we could do this in a lot of different diseases with a lot of different constructs, and you reproducibly get the same outcome, which would be correction of the genetic defect in a variety of diseases. And it might take a number of years to get there, but I don't think it's totally it's not totally science fiction. It's probably science fact 20 or 25 years from now, as you first get enough experience showing that these editors can be used across the entire set of mutations. Then you feel comfortable that the principle can apply across different diseases, and that you feel confident enough that the targeting systems go to where they need to go to, that this can become essentially almost an AI-driven affair, to pick an appropriate guide and an appropriate delivery vector for a given entity. And it's a great way to end the day at kind of it's been a long day, so it's kind of in this haze of science fact and science fiction. But I do think it's not so fictional, because I think it is possible that that's where this could go, particularly because unlike our current gene therapies that require us to grow up cells, etc., here you could imagine that this is synthetic. This is basically synthetic biology that could it could be leveraged to complete out a product. Does that have I lost
I
my
think
mind?
that,
or is
no, I
Yeah.
appreciate you saying that it's not a completely crazy idea. Thank you for that. And I think this actually came up. I'm involved in a lot of these discussions in the context of oligonucleotides and in a different conversation. Could that ever become the practice of medicine? And so that's where the idea is, could we apply it to genome editing? But I really appreciate your comment, and that it is something we can imagine. I realize obviously this will be way far in the future. Right now we're in the midst of drug development in very early days, but that's kind of why I wanted to just bring up this kind of blue sky idea. And if you have any thoughts about your vision of where the optimal future would be, I'm sure we'd love to hear it. But
no,
totally
I
up
think,
to you.
I mean, I no, but I think I think where this would go is I think where we may be headed is in a place where whether you consider this practice of medicine, or whether you consider it a manufacturing device that is enabled with AI to produce genome correcting products, I think that may be the place that this goes ultimately, if you were able to show that all the algorithms could get you to the right place. There's a lot, just in case anyone who's listening from the press or anything like that, there's a lot to get from here to there that's years away. But you know, I'm always impressed that, you know, sometimes you have to be visionary, because there was an author who back in about, I think it was something in the mid to late, it was probably the late 70s, was talking about the linkage of the population with these personal communicators, that, you know, we're going to allow everyone to have information ready at the fingertips. And I think back in the late 1970s, early 80s, that sounded like totally, you know, crazy that everyone was going to have a personal communicator, particularly in the mid to late 70s. And now, look at what we're doing. So I think this is, again, you know, the kind of the visionary look to where this might go. I think it's pretty impressive.
Right. Thanks, Peter. Sounds like a great way to end. Yes, I would agree.
Thank you both. Peter, thank you again for taking the time to join and kick off our Meet the Expert series within the SCGE. We really appreciate you, the participation, and the discussion that we had, and all the participants that joined the panel, and all the great discussions that we had around that. So thanks, everyone. Mindy, I don't know if you have any closing remarks.
Nothing too much, just that we will capture all of these questions that weren't answered and share them within the consortium. We might use some of them for cross-cutting discussions coming up, and try to get answers back to the consortium members for questions that weren't able to get to today. So thanks everyone for coming. Thank you, Peter.
Thanks so much for having me, and thanks for letting me speculate wildly here. But we'll try to actually make some of this into reality in the not too distant future. Thank you.
Wonderful.
Thanks, Peter. Thanks, everybody.
Thanks, everyone.
Thanks.
Bye.